That's all we did. And then once we decided to kill things, yeah, you do the skin transplants. The skin transplants. Oh, the skin transplant won't matter this time. You look at the I decided not to shoot.
Should be under the modules. I think helpful.
No,
Helpful information.
Yeah. Because it's short answer. Oh, that's really wrong. And he already took out all the easy bacteria.
All right. It's enough time to fight technology. Okay. So what do I want to talk to you guys about today? We're going to build on our discussion from last time, moving on from adhesion to talking about the actual process of invasion through host cells. Shouldn't be crossed out, but what we're going to do first is talk about invasion of the intestinal epithelium. So why are we going to focus on that? Well, the reason we are is because understanding gastrointestinal infectious disease is where we learned the vast majority of what we understand about bacterial pathogenesis, the interaction of pathogens with their host, and what the host is doing in response to pathogens and essentially managing the microbial population that resides in your intestines. So remember, the intestines are essentially the most abundant numbers and diversity of microorganisms in our body are in our gastrointestinal system. And remember that epithelium is essentially the type of tissue that is lining your gastrointestinal system. And I'm going to talk to you about two different model systems. One that involves what's known as a trigger mechanism for invasion of epithelial cells. The other is what's known as a zipper mechanism for invading epithelial cells. And I'm going to use two different model bacterial pathogens, gastrointestinal pathogens, as a backdrop for giving you kind of the fundamentals of what's going on with these two modes of host-cell invasion. And there's also a reading for today that will also take you through those examples in much more detail than I'll take you through today. And we'll end today talking about how you actually study this outside of an animal. But before we get started on that, I wanted to touch on something that is in the syllabus. It is part of your grade, and what's going to happen is you guys have to give a presentation, and you'll be assigned to a group of 3 or 4 people. I do the assigning, and so we're going to work with two or three other classmates, and what you're going to do is basically take us through the infectious life cycle of a particular pathogen of your choosing. So things that you can't discuss are listed here because these are essentially the pathogens that I'm going to be using to explain these concepts to you, and I have been so far. So thinking about Salmonella, Listeria, Vibrio cholera, E. coli, Neisseria, or Shigella, they're off the table. Okay? So there's lots and lots of other pathogens. So remember, first day, I told you, there's, let's say, like 100 important human bacterial pathogens. There's going to be like 10 groups in here. So I think you guys can manage to find some other pathogens other than these ones. So what you're going to do, I'm going to assign you your groups over the weekend. By October 12th, I think I've given you guys ample time to decide on a pathogen. And so the way that I deal with this is first come, first serve. There'll be an active list on Canvas. So as soon as your groups contact me and say, hey, we want to do my favorite pathogen, I will add that pathogen to your group on Canvas. So you guys will have sort of a live, real-time list of, shoot, group 3 chose our pathogen. Let's think of a backup. So if you're wanting to get inspiration for pathogens, I encourage you to look at the science section of the New York Times, Washington Post, USA Today, sort of any reputable resource like media source. Look at their science section, and I guarantee usually about once a week there's going to be some infectious disease related article talking about some interesting pathogen. Or if you know something that's not on this list that you cannot discuss with items, talk to about your group. So by the end of October, you'll notice that these are in about 2.5 week increments. So I'm giving you plenty of time to decide on a pathogen. I'm giving you about 2.5 weeks to think about, all right, what can we dig up about our pathogen that we've chosen in terms of these aspects of their life cycle that we'll talk about in a second. And you're going to have to provide me an outline by Monday, October 29th. And then right before, basically the Friday before Thanksgiving break or the fall break, I'm going to need from you a draft of your presentation. So I'm going to give you feedback over that weekend. And since this is essentially, you're going to be using Google Slides to do this, so you can all work on this together in real time. And why I want this draft done and why I want to get you feedback is because presentations begin Tuesday, November 27th. So the last 4 lecture days are slated for you guys to give your presentations, okay? And so what I can't cover in terms of breadth in different pathogens, this is what you guys are going to be doing to teach each other about a lot of other interesting pathogens than the ones that I share with you in class. And you become your own expert in a particular topic. So how does this break down? So there are all of these pre-presentation deadline materials. So I'm giving you waypoints, right? So nothing is all due at the end of 5 or 6 weeks. What's going on is that 35% of your grade is essentially sticking to those deadlines that I've given you and doing a good job, right? So take this seriously, and that 35% is in the bag. So your presentation, I will score your presentation based on its content, organization of concepts that I'm going to give you in terms of a rubric, and the clarity of this presentation. So I'm giving you how I'm breaking down how I'm assessing you. And also, you all have to sit and participate, and you have to listen to everyone. And so 15% of your grade is going to come from your classmates' assessment of your presentation. So this is a group assignment, right? And so I'm not ignorant to having dealt with group work myself. And there are group members who pull a lot of weight, and there are group members who don't. Ideally, you want an equitable division of labor. And so what happens is that your group is given an overall score for this presentation, and I scale that score based on the individual in each group based on your group's assessment of you. Okay? So what does this mean? If you're a slacker, you're going to pay for it in the end. And ideally, this should not be shocking to you if you put in very little effort into a presentation and you get a bad grade, right? So be equitable in your distribution of labor in your groups, and you don't need to worry about that, right? So, that said, are there any questions about this presentation? So, based on the number of groups and the available class time, Oh, come on. I worked through this. So it'd be around like 15 or so minutes. And so 15, 20 minutes. I think you're right. Yeah. So I'll put up more information on Canvas about what my explicit expectations are about this. But I want to get your gears turning. So when I assign you your groups this weekend, you guys can immediately kind of huddle and decide, this is what we want to do, right? And so part of why I'm giving you 2 weeks to make a decision about that is it behooves you to do a little bit of legwork and say, like, how much do we know about this pathogen? Because as you'll see in the rubric that I'm going to give you is that there's no penalty in picking a pathogen that we honestly don't know a lot about yet. So finding something that's an emerging pathogen, that's challenging, right? And so outlining what it is that we do know is just as important as being able to identify what we don't know. And given how a particular pathogen, what it does, you might be able to tell us, like, these are the sorts of experiments that you want to do to try and understand this. And we can do this because we can culture this thing, or we can't because maybe we can't culture this bacteria, right? So, that said, are there any other questions? Yeah.
So pathogens include bacteria, viruses, parasitics, and fungi. Because this is a bacterial pathogenesis class, would you rather us just stick to bacteria, or are viruses okay too?
What do you think?
I'm going to say just bacteria.
Yes.
Okay.
Yep. All right. So since we are topically restricted to talking about bacterial pathogens here, we're going to focus on bacteria for these presentations. Are there any other questions? All right. I'm sure there will be more soon. But just to prime your minds for what's to come. All right. So what are we doing today? So again, we're going to talk about invasion of the intestinal epithelium and methods for studying invasion and spreading. So back to our roadmap. Do you guys prefer these lights on or off? On? Off? Okay. All right. So our roadmap. So what did we talk about last time? We talked about adherence to skin and mucosa. And where are we going today is we're going to deal with this middle box right here. Okay? And we are going to talk about invasion of epithelial cells. But before we do, I think it's important to understand the normal course of events that are going on with epithelial cells in epithelial tissue in your intestine. Okay? So, in and amongst the epithelial cells in your intestine, and so what this cartoon is showing here is a monolayer of these epithelial cells that are all stacked together and forming this epithelial cell monolayer that lines your intestines. Okay? And again, we're focusing on our intestines because this is one where the part of our body that does the most immunological sampling of anywhere on our body. So there are a massive number of lymph nodes lining that are basically sitting underneath these epithelial cell layers, and in lymph nodes are a whole bunch of B cells, T cells, all of these adaptive immune functions that we talked about. So something that we're constantly doing is surveying what's going on in the lumen, essentially the place where all of our food and liquids that we consume are moving through our intestines, right? That what's going on out in that lumen, we are constantly sampling what's going on. And it's an interesting process in how this happens. And so the things to keep in mind is that these epithelial cells that line our intestines, the vast majority of them are non-phagocytic. Okay, so they're not naturally taking things up by endocytosis. However, there are a family of very special epithelial cells that are called microfold cells, or M cells. And what these are is that they are naturally phagocytic epithelial cells. So they're essentially developed differently than your normal run-of-the-mill endothelial epithelial cell, in that they engage in active uptake of things from the lumen. And so they're basically swallowing and making vacuoles of stuff that's out in that mucosal layer surrounding the surface of these epithelial cells. And what we know about them histologically, why we know that they're different, is that they're not microvilliated. And so in this cartoon, you see these epithelial cells have these little like finger-like protrusions. And so essentially what that surface of those endothelial cells in the epithelium are doing with those like invaginations and basically making these little dendrical fingers is that they're maximizing the surface area of those cell surfaces for mass transfer. And so taking up nutrients out of the lumen of your small intestine, there's basically those surfaces are chock full of transporters and all kinds of things in epithelial cells. However, M cells are relatively smooth. And they're much larger in terms of their sort of physical footprint. And they're constantly gobbling things up from the lumen. And what is shown here in this cartoon are these spiky-looking purple cells. You might be saying, oh, what the heck are those? Those are dendritic cells. And so if you remember, dendritic cells are antigen-presenting cells, okay? And dendritic cells and M cells are essentially, they're kind of married to each other in this basically just underneath this epithelial layer here. So M cells are surveying, they're bringing things up in vacuoles, they're essentially spitting stuff out to these dendritic cells, which then naturally take these things up, process what's been taken up, and present it on MHC, or major histocompatibility complex class 2 receptors, right? And they're presenting antigens. And what's also present underneath this epithelial layer are T cells and B cells, and they're essentially waiting to be presented things all the time by dendritic cells that are sitting just underneath these M cells. And so we're constantly surveying what's there. And so you'd imagine what we end up seeing with infections, with pathogens in our gastrointestinal system, is why this system works so well, is that if you have a population explosion of basically the wrong kind of bacteria, we're constantly sampling. And if you're sampling randomly, you're just going to sort of build random immunity to various things. Now, if you are exposed to a massive amount of, say, Listeria or Salmonella in the lumen of your intestine, those M cells are going to be taking up Listeria cells, for example, dumping those Listeria cells down into these dendritic cells. These dendritic cells are going to grind them up, present antigens, and you're going to build immunity preferentially to those things that are at the highest abundance in your GI tract. Okay? So if you have to distill all of this down to two things, is one, the vast majority of cells in your epithelial tissue, these enterocytes, they are non-phagocytic. So they're not taking things up. They're just basically a physical barrier between what you eat and the connective tissue underneath in your in the vascular tissue and lymph tissue that's underneath in your gastrointestinal system. And that these microfold cells, they're essentially kind of a sentinel. They're constantly taking things up and presenting different things from our diet that we've eaten, that are residing in our mucosal layer nearby. They're presenting that to our adaptive immune system all the time. And so you can imagine why we start thinking about these sorts of interactions when we think about things like Crohn's disease and various other autoimmune disorders that are so difficult to understand, is I can give you this very simple, nice, cute picture, but in reality, this is not everything that's going on. So this is incredibly complex, and you are essentially sampling from the environment and feeding things into your adaptive immune system all the time. And if you're generating immunity against the wrong things based on what is being presented, you can have all sorts of problems. So, that said, what does this physically look like? And so what I'm showing you here is a histological cross-section of a part of the ileum of the small intestine. And so the ileum is part of your small intestine. And it's actually where the highest density of lymphatic tissue is in your gastrointestinal system. And it is primarily what you're seeing here, these stained things here. These are all essentially mesenteric lymph nodes. And above them are what are known as these Peyer's patches. And this is essentially the physical picture of what that cartoon was talking about. And then these, here, these are your normal enterocytes. And you see they look really rough on this when you perform SEM imaging. And those are all of those little fingers, right? And so the surface area, the available surface area for mass transfer in this particular arrangement is extremely high. Now, what do you guys think this is? It's an M cell, right? So I told you they're far smoother and they're fairly large. And what they're doing is essentially these ripples here are more or less like beginnings of forming endosomes and vacuoles that are just taking things out from the environment all the time. And so this physical arrangement of these M cells sitting here, and what all of this is, these invaginations, so these are the villi that are in your gastrointestinal tissue, and what is going on here is essentially you have layers and layers and like basically just up and down, up and down, up and down of these epithelial cells, these enterocytes lining this. So at multiple levels of organization, you're trying to maximize surface area in your intestines. So both at the cellular level, in terms of how much membrane surface area there is, as well as how much epithelial tissue is actually being presented to the outside world. And so, where you often find these M cells are down in what are known as crypts, so these kind of crux in between villi in your intestine. You'll find a higher density of M cells there, and that these are essentially then interacting with this Peyer's patch, which is filled with B cells, dendritic cells, and basically all of those adaptive immune cells that we're talking about. And underneath this is a lymph node, which is packed with B cells and T cells as well. And essentially, once certain B cells proliferate after being activated by the adaptive immune mechanism that we talked about prior, you essentially get a whole bunch of plasma B cells, and those lymph nodes basically allow spread of those antibodies and those plasma cells all throughout your body through the lymphatic system. So you can end up with systemic adaptive immunity to things that you are essentially sensing in your gut, which is pretty extraordinary. Okay, so this sets up an interesting issue in that what's good about this is that we're sampling all the time, right? The bad part about this is we're eating all the time, and while we now have a fairly sanitary diet, the vast majority of human history is not, and so we're constantly bombarded with different things from food, for example, and this high surface area does wonderful things for us in terms of mass transfer, and us being able to efficiently extract nutrients from food, but it also presents a really extraordinary environment for pathogens, right? That you have a massive amount of surface area to interact with as a pathogen. And as we're going to see in some examples, both today and in the future, you have essentially a mechanism that we talked about this epithelial cell layer as being a boundary, right? That this is this kind of physical gatekeeper between the outside world and the rest of you, right? Now, I just told you that there are special cells that essentially, like, oh, I'm just gonna take up everything and then dump it down to these dendritic cells, and they'll grind it up, and we'll generate adaptive immunity this way. But this is essentially a really bad thing if a pathogen is able to deal with this, right? So if you can survive the intracellular environment, you can survive in dendritic cells. We are essentially are the creators of our own demise in some ways, and that, you know, we're taking things up. We're providing a highway for pathogens to bypass this epithelial cell layer. And so some pathogens that we'll see, Shigella is one that exploits these M cells. Salmonella does as well. But I'm going to talk to you about a more generic mechanism that a lot of pathogens, gastrointestinal pathogens, use. And that is, while these M cells are around, they're fairly rare. And if you have the ability to circumvent or utilize normal epithelial cells, enterocytes, to get across that epithelial cell layer, now you don't have to be searching for special cells. You can just interact with any old enterocyte and gain access to everything that is underneath these epithelial cell layers, right? So there's a lot of connective tissue down here. There's a lot of other things going on, a lot of nutrients, compared to what's in the lumen, and especially in the mucosal layer that's right above these epithelial cells. So before we get into mechanisms, are there any questions? Any questions? So have you guys heard of microfold cells before? No? Okay. Yes, and everyone else, I'm sure, is what the heck is this guy talking about? But I'll inundate you with this as the next couple lectures go by. So you as a pathogen, you want to get across that epithelial cell layer. So as I mentioned before in the last lecture, there are kind of these two fundamental decisions that a lot of pathogens are going to make. One is, am I going to be an effective extracellular pathogen? When I say that, what I mean is that I can outcompete native flora, and I don't need to engage in trying to transverse this basolateral membrane underneath these epithelial cells and get into connective tissue to grow and thrive in a host. What we're talking about today are pathogens that make the decision that, you know what, like, I'm going to engage in an intracellular lifestyle, or I'm going to use this intracellular lifestyle to be able to gain access to things that are beyond this epithelial cell layer. And we'll get into more detail in the next lecture about some of this. But what I want to get across today are two key mechanisms that we understand well about how bacterial pathogens basically reprogram normal enterocytes to be phagocytic cells. So what I was getting at before is that, you know, say one in 10,000 cells in your intestine might be one of those microfold cells, and all the rest, by and large, are enterocytes, right? And so if you have some mechanism to make enterocytes do what microfold cells do, you can both inhabit those enterocytes, as well as get those enterocytes and potentially move you across that epithelial cell layer. And if you are essentially moving across through an illegitimate pathway, moving through enterocytes and not microfold cells, the probability of there being a dendritic cell waiting there to eat me on the other side is also lower. And so there are some advantages to using enterocytes, both in terms of probability, so like, hey, I can show up, I can get anything to do what I want, as well as what happens on the other side of that basolateral membrane. So I'm talking about two mechanisms, the trigger mechanism and the zipper mechanism, and there's sort of a spectrum of things in between, but these are the two fundamentally most important that I would want you to understand from a course like this. So, what these two mechanisms fundamentally are doing, so they're essentially performing the same function, that you as a bacterial cell want to interact with an enterocyte, and I want that cell to take me up in a vacuum. And what happens in this trigger mechanism, as we're going to see, is that this is a process by which a bacterial pathogen is using what are known as effector proteins. And what these effector proteins are essentially bacterial proteins that get secreted directly into the host cytoplasm and mess with particular host signaling pathways. As we're going to see, a lot of them, these pathways have to do with actin polymerization as well as microtubule formation. And both of these things are important for endocytosis, right? So being able to basically ruffle your membrane and take something up is the result of localized actin polymerization. And essentially, pathogens like Salmonella, what we're going to see is that they use specialized secretion systems like we just talked about a couple lectures ago to deliver their effector proteins from their cytoplasm directly into the host cytoplasm, which initiates this host response that is membrane ruffling and forming an endosome, which is again something enterocytes do not normally do. So if Salmonella interacts with those, binds to and delivers its effector proteins into enterocytes, enterocytes will engage in endocytosis and take Salmonella up. Now the second mechanism that we're going to talk about is the zipper mechanism. And this is the mechanism that Listeria uses. And what this mechanism is this uses these adhesins, these afibrillar adhesins that we talked about before, except this is a gram-positive pathogen. And so it is basically anchoring those adhesins to the cell wall instead of sitting in the outer membrane. And that these are going to interact with specific host surface receptors that are responsible for a variety of different signaling. But for Listeria, one of the important things that it's going to bind to is decadherin. And so essentially what it's doing is interacting with a whole bunch of host receptors that are normally responsible for saying, hey, I'm touching another host cell, and I need to form cell-cell junctions. And what these bacteria are doing is interacting with multiples of these cadherin receptors and inducing endocytosis through interaction with multiple surface receptors and basically formation of this vacuole. So the output of both of these processes is the formation of a vacuole that this bacterium is now residing inside of the host cell. The trigger mechanism uses secreted proteins to perturb normal signaling in the host cell to induce endocytosis. The zipper mechanism does not use effector proteins. It uses surface proteins on the bacterium that are going to interact with host cell receptors that stimulate actin polymerization and microtubule formation that are both required for forming an endosome. So in both cases, you form endosomes with bacteria in them, just whether or not they're using effector proteins or just interacting with host surface receptors. So, are there any questions? Yes?
I'm sorry, I don't understand. So endosome and vacuole, same thing?
Yeah.
Okay.
Yeah.
Is the zipper mechanism only in with gram-positive bacteria?
So it is not only in gram-positive bacteria, but it is most prevalent in gram-positive pathogens. Yeah.
Zipper mechanism to interface the host surface receptors. What do you say the trigger mechanism is? They insert.
So they deliver what are called effector proteins. We'll see what some of these effector proteins do in a minute. But if you remember, there were 3 of the 6 specialized secretion systems we talked about that are able to deliver proteins from the bacterial cytoplasm into the host cytoplasm, right? So the type 3, type 4, and type 6 secretion systems will do that. The most prolific in bacterial pathogens that gets this job done is actually the type 3 secretion system. So it's basically a molecular hypodermic needle that is structurally related to the bacterial flagellum. And in gram-negative bacteria, they make a lot of bacterial pathogens, gram-negative bacterial pathogens will make these type 3 secretion system needles, which essentially stab into the host's cytoplasmic membrane, and you have this essentially molecular straw that is going to pump these effector proteins that are produced in the bacterial cytoplasm into the host cytoplasm, and these perturb a lot of different host cell processes depending on the pathogen. And so, yeah.
So are the effector proteins, are they breaking down the actin and like changing the shape of the cell so that the bacteria will be taken in, or is that happening by way of the neocomplex?
So the effector proteins are what are doing these things.
So they're like breaking down the structure of the cell.
Yeah, or they're causing large scale changes in how the cell is managing where actin is being polymerized and depolymerized. Yep. And so, how do we know that? Well, we can heterologously express effector proteins in enterocytes in tissue culture, and we can essentially make a large number. You can get these enterocytes in tissue culture to start displaying some weird abnormal cell behaviors depending on what effector proteins you're expressing. And so Salmonella, conservatively, it's around 20 so effector proteins that it'll produce depending on the isolate. And there are other bacteria, so Legionella pneumophila that I talked about last time. Since its normal life is interacting with amoeba and getting amoeba to do this, and basically reprogramming amoeba cell biology. They range, depending on the isolate, anywhere from 200 to 1,000 effector proteins that they'll produce. And so you have this massive diversity in effector protein production in different pathogens. Salmonella are really hardcore animal pathogens. And so many of the effector proteins that are produced by Salmonella will elicit the same cellular response in a wide variety of different animal cell types. So, boiling down what I said, the bacterial factors involved in trigger versus the zipper mechanism is that this trigger mechanism absolutely requires secretion systems. Specifically, it requires the secretion systems that are capable of delivering proteins from the bacterial cytoplasm into the host cytoplasm directly. Okay? And so this is going to be one big functional difference when we start talking about exotoxins, okay? And so there's going to be some similar mechanisms that exotoxins exploit, but they're fundamentally different from effector proteins, and then effector proteins are utterly reliant on being delivered directly into the host cytoplasm, whereas exotoxins can be secreted out into the environment, and that they have sort of their own delivery mechanism for getting into host cells that we'll talk about later. The zipper mechanism, this is what these afibrillar adhesins are in many cases doing. That they're really high affinity interactions, and that they're targeting very specific host surface receptors. Does actin polymerization occur in both cases? Absolutely. And so it just comes down to how do you get that to happen inappropriately. So the zipper mechanism is essentially I'm going to interact with a whole bunch, or I'm going to interact with host surface receptors that would normally be interactions would normally be triggered by cell-cell contact, for example, between two enterocytes. So this listeria is going to come in and interact with these same receptors and essentially fool these enterocytes into believing that they're interacting with another enterocyte and begin essentially formation of what would normally be kind of a cell-cell junction, except there's no other cell there, so you end up forming an endosome instead. And so how do bacteria enter? And so in the case of this trigger mechanism, it's really loose association with the membrane and membrane ruffling. And so what we're going to see is that injection of these effector proteins, if you're Salmonella, into the host cell is going to perturb a whole bunch of things about how actin is being polymerized very proximally to that surface, the membrane that's essentially touching the lumen of the intestine, right? And so Salmonella is pumping these effector proteins in, perturbing cell signaling. And what you see, if I were to express a number of these effector proteins in enterocytes in tissue culture, is that their surface morphologies would change drastically. So they begin forming these large ruffles, kind of like what M cells do, and that they're just going to sort of illegitimately start forming vacuoles as a result of this membrane ruffling. And so what the end result is that the endosomes or vacuoles that get formed via this trigger mechanism are much larger than endosomes that are formed by the zipper mechanism, which is through tight association with the host cell membrane, that then you basically form this really tight cocoon around the invading bacteria. So are there any questions? How many of you guys knew that bacteria did this? How many think that that's pretty crazy? Like half of you guys? Yeah, no, I mean, the thing that's frightening and like astonishing about this is that especially this trigger mechanism, that these are bacterial-derived proteins. These are proteins that evolved for millions of years in bacteria to serve other functions, and were co-opted and have new use and reprogramming eukaryotic cell behavior, right? And so that to me is really nuts, right? That we're talking essentially across domains of life communication. That these pathogens, that they've been under such intense selection for exploiting the host that this is what's emerged, right? That there's these couple of fundamentally different ways to get this job done, but both are highly effective. And the craziest one to me is this trigger mechanism. Right? You're relying on a protein secretion apparatus that's going to deliver proteins from your cytoplasm as a bacterial pathogen into a host cell. And if those proteins that I shot into this host cell are going to mess very specifically with specific host cell signaling pathways, they're going to elicit a response that's beneficial to me. Right? So I mean that to me is absolutely mind-bending. So let's get into some nitty gritty. So I kind of give a high level picture of what goes on with some of these guys. So I told you I was going to give you two examples. One, Salmonella enterica, the other, Listeria monocytogenes. And Salmonella enterica is one of my favorite bacteria. It's an excellent model for both bacterial genetics, molecular microbiology, you name it. We've learned an awful lot about how biology works by studying Salmonella and its close relative E. coli. And it's also extraordinary in that essentially everything that we learn in E. coli or in Salmonella, we get insights into pathogenic behavior of the bacteria. So all of the extraordinary molecular microbiology and genetics and all the cool tools we've developed to do things in E. coli and Salmonella for other purposes, or just understanding how various molecular mechanisms work, we can bring all of those same tools to bear in understanding how this bacterium interacts with its host to elicit a pathogenic response. Okay? So it's a gram-negative rod, and it's found in the intestines of many vertebrates. And so there are various species of Salmonella that are commensals to various avian and reptile hosts. And in fact, Salmonella enterica is often found in the GI tracts of turtles and other reptiles. So that's why one of the reasons you're told, if you're handling reptiles, wash your hands after you're done, before you go eat and drink things, because your chance of getting Salmonella from contact with reptiles is pretty high. So it's not a natural part of the human flora, and we'll see some of why in a second. And so how is it transmitted? Contaminated water, that's sort of the number one source throughout the world. If I were to say, how are you and I going to get salmonellosis or salmonella infection in the United States, is through eating uncooked or unpasteurized foods or beverages. Also emerging are problems with vegetables. And so some of this is because of the use of livestock feces in fertilizing crops, for example. And it's turning out that we're understanding that Salmonella is actually really good at interacting with plants and getting plants to do certain things to benefit it and essentially survive on plant surfaces. And some of the same mechanisms that are important for invading us are also important for interacting with plants. And the last is exotic pets, right? So if you have reptiles, wash your hands after you handle them. And so there are actually a wide variety of diseases that are caused by Salmonella enterica, and they're very, very serovar or serotype dependent. And what we see when we break down Salmonella enterica below just species, and looking at these serotypes, you'll see all these different things like Arizonae, Typhimurium, and Enteriditis. Sometimes this has to do with where this Salmonella is isolated in terms of an outbreak. Other things are, so Typhimurium, this is the serotype that I work with in my own research. And so what Typhimurium, if you were to say, I work with a murine model, what does that mean?
Mouse.
A mouse, right? And so, and typhi is essentially typhoid fever. And so this particular serotype will cause typhoid fever in mice, but in us, it causes gastroenteritis. And so it's a localized, self-clearing infection. And so the three that you would most commonly get infected with in the United States, if you're into hanging out with reptiles, chances are you will at some point get an Arizonae infection. The two that are most common in the United States are Typhimurium and Enteriditis. and you're going to just basically get nasty, self-clearing diarrhea for 7 to 10 days. And so if you're healthy, you're gonna be fine, but you will be miserable. Where it becomes life-threatening is if you are immunocompromised or sick with something else. It can be really dangerous. Where things get much more nasty are Coleraceus, Dublin, Paratyphi, Typhi, and these sort of down this list increase in terms of their nastiness to us. And what the big transition is, and this will be our second paper that we're going to talk about, is that what these serotypes have acquired are mechanisms to move on from just being a localized infection, but you can become a systemic pathogen. So using the same invasion mechanism we're going to talk about, so the Typhimurium in us would just hang out in those mesenteric lymph nodes and stay there. These other guys, what they do is that they are much more aggressive, invasive pathogens that are going to move on into other places in your body and cause much more severe problems, whether this be typhoid fever, for example, or septicemia. So this is, I think, our first glimpse into this idea that one species of a pathogen doesn't necessarily dictate exactly what disease symptoms you're going to get from that specific pathogen. There's a wide variety of things that the same core pathogen can do with a few extra tools in its tool belt. So we'll talk about this when we start talking about horizontal gene transfer. Yeah.
Do we need to know all these?
You need to know all of them. Well, you need to know everything that I say. No, I'm not going to ask you about serotypes, but I want you to be able to think about this idea that one serotype of the same species would just give you bad diarrhea, and another could give you typhoid. Essentially the same pathogen with just a few subtle differences. Yeah. Okay. So the course of infection for Salmonella is that it invades intestinal epithelium, primarily in the ileum of the small intestine, where we have the highest abundance of essentially these Peyer's patches, M cells, and the surface area of the ileum is very, very high. So the bacteria, once they invade epithelial cells, they replicate in endosomes. So those little, essentially the vacuole endosome that they make through inducing uptake, that becomes their home. And they essentially use epithelial cells to then gain entry into macrophages on the other side of the epithelial cell layer, and basically hitchhike in macrophages to gain access to the lymphatic system and blood vessels. And so normal Salmonella stops, or Typhimurium stops in mesenteric lymph nodes. So it just stays there until it's cleared. And we'll see some of why a little bit later. But say Paratyphi or Typhi, they're going to essentially use your lymphatic system to spread throughout your body. Okay? And this is where you can end up with distal infections in other organs and tissues as a result of essentially invading in your intestine and giving you septicemia. Okay? So, much of what we've seen, one of the critical things, of course, is adhesion, and Salmonella uses fimbrial adhesion to stick to enterocytes. It also appears that they use flagella to be even more sticky, and once stuck to the surface of enterocytes, they will then produce a type 3 secretion system, shown here with the little orange rectangles, and then they will inject or secrete effector proteins into these enterocytes, which induce membrane ruffling and uptake into a vacuole, and essentially internalization into epithelial cells. Yes?
So does Salmonella choose to attack enteric cells because there's no dendritic cells in there? Why does it choose to go through enteric cells whenever there's an end cell that's going to readily absorb it?
So it will also enter through M cells, but it will also use enterocytes. So it just basically, it's any way in, it will do it. But it's really good at doing this with enterocytes. And so if you have the tools to essentially get normal cells that don't engage in endocytosis to begin the process of endocytosis, now you have access to a whole lot more ports of entry into your host than relying just on M cells. Yep, good question. Great question. Okay, so I talked about these effector proteins. They get secreted from the bacterial cytoplasm into the host cytoplasm. And what do? I'm going to give you some examples of some here, and I'll give a sort of pictorial description. I have a video. Didn't show. So what's going on here in this little movie is you can is this idea of membrane ruffling. So this is a Salmonella cell that has interacted with an enterocyte in tissue culture, and this membrane ruffling is the result of, so when I was talking about membrane ruffling, it's essentially the membrane going nuts and forming an endosome and then stopping. And so what we saw in this picture was that this is a time lapse, right? And so, I mean, it moved pretty quickly. This process doesn't happen quite that fast in real life. But what I want you to take away from that video is that there had to be a process of saying, all right, membrane, go nuts. And then a process of saying, okay, stop doing all of that. Go back to being like you normally were. And so how this is mediated is through the action of these effector proteins. And so we'll get as far as we get today, and I'll finish our discussion next time. But there are a number of effector proteins that get secreted. So I'm giving you some examples here. So SOP-E, CYP-A, and SPP. And so what these do is through interaction with various host signaling factors, what I want you to be able to think about is that you want to stimulate actin polymerization, and you also want to inhibit it, right? So there's a period of time that you want to say, like, all right, host cell, go crazy, take me up, and then you want all of that to stop, right? And so there's competing things happening here, that you have effector proteins that stimulate actin polymerization, and you have ones that inhibit depolymerization. Sorry, and then you have other effector proteins that turn this on. So what goes on then is that in this diagram here, Salmonella interacts with an enterocyte, injects these sort of major effectors, CYP-A, SOP-E, SOP-B, SPP. And so SOP-E and SOP-B, they are essentially messing with RAC signaling, which is important for intracellular trafficking and actin rearrangement, and essentially are basically locally saying, all right, actin, start polymerizing, and the mechanisms that are around to say stop polymerizing or depolymerize, you inhibit those, and so you induce this ruffling, through basically overstimulating actin polymerization and taking the brakes off of actin polymerization, which is depolymerization. So you polymerize actin, and it causes actin to just get long and forcibly make these ruffles. And then the SPIT-P effector essentially encourages transition of this phosphorylated version of this RAC signaling pathway to essentially convert back to its inactive state, which is this GDP RAC1 CD42 complex. And so basically what is going on is in the early stages, the thing that I want you to keep in mind is that it injects all of these effector proteins essentially at once, right? And so the way that this whole process gets mediated is that the activity of SOP-E and SOP-B is much, much higher than SPIT-P. And so you preferentially phosphorylate the signaling complex. And CYP-A is also all the while inhibiting actin depolymerization. So you over-polymerize actin. Now the thing about the protein half-life of SOP-B, SOP-E, and CYP-A in the host cytoplasm is they're much shorter than SPIT-P. So SPIT-P has a much longer protein half-life, and so after these guys all get degraded, now this whole mechanism gets shut back off. It essentially returns to a state that looks like, for the host cell, like, wow, I totally freaked out there for a second, but I'm cool now, right? So there's this really elegant co-opting host signaling, get what you want, and not only just saying, like, I turned all of this on, but then you also have a mechanism to essentially turn it back off, right? And so some of the reasons for this is that if you're Salmonella, you want to invade this host cell. And as we're going to see, I think in the next lecture, if you do this wrong, all you're going to do is tell this enterocyte, you know what? I'm going to engage in apoptosis, right? And that is essentially, for Salmonella, that doesn't really do you any good, right? As we're going to continue to see about how Salmonella's life cycle and what it does. So being able to invade your host and essentially say, like, you had this momentary freak out, everything else is all okay, right? And as we're going to see, once pathogens exist in these intracellular vacuoles, they start to do other things to elicit different behaviors from these host cells. We'll see next time. All right. Are there any questions about Salmonella? All right.
Should we know what each of the effector proteins do?
Should you know what each of these effector proteins do? I'm not really big on memorizing names of things. And the bigger things that I want you to be able to take away is that, one, you can inject a whole bunch of effector proteins all at once. And the way that most pathogens bias host behavior is not through selective, like, well, you know what, I'm going to send over this effector first and then wait for something else and then send the next one. But essentially, you're just going to dump all of these effector proteins in, and through differential activities, so say like SOP-B and SOP-E, that they're really good at getting this signaling pathway activated, right? So they get it phosphorylated, and simultaneously, the SPIT-P protein is also there, encouraging dephosphorylation of this same signaling complex. But these guys are just much better at encouraging phosphorylation, so you bias this thing towards being phosphorylated. And simultaneously, CYP-A is saying don't depolymerize actin anymore. But then this idea that if you provide all of these effectors that are going to modulate host behavior all at once, that it's through differential protein half-life in the host. That's how But that's how the original kind of like, all right, start membrane ruffling, how that gets turned off is through those top three effector proteins getting degraded faster than plastic. So am I gonna, do I like saying like, what does SOFT-E do? No. But conceptually, because I mean, you could replace those with anything like, the most important thing is this concept of how do you elicit a behavior from the host and also turn it off, right? Okay. So we have a few more minutes, and I'll introduce Listeria, and we'll get into the nuts and bolts of what it does next time. So I wanted to contrast Salmonella with a fair bit less known pathogen, but it's an important intracellular pathogen, both in the fact that it invades host cells, but it engages in an entirely different intracellular lifestyle that we're going to see next time compared to the Salmonella does. So Listeria monocytogenes, it's a gram-positive rod. It's found in soil, water, vegetation, intestines of animals. It's another one of these like, it's found everywhere sort of bacteria. And what's really interesting, and there are not quite as many humans that are asymptomatic carriers of Salmonella, but there's a fairly sizable amount of the human population that are just asymptomatic carriers of Listeria monocytogenes. And one of the important things about it is that it causes inflammatory diarrhea. It's also an invasive pathogen if you are immunocompromised. It can grow at cold temperatures. Salmonella is not necessarily real good at growing at, say, 4 degrees centigrade. This guy is. And its association with agricultural crops, so melons, for example, are a common environmental place that they inhabit in terms of vegetation. And so they will continue to colonize vegetable surfaces at cold temperatures. Additionally, in terms of food processing, they are tolerant to a lot of preservatives. And so, like lunch meats, for example, are another place that you'll find Listeria. And one of the interesting things about Listeria is it's one of the few bacterial pathogens that if it becomes a systemic infection, passing, like moving on beyond your gastrointestinal system, for women, it's actually the only bacterial pathogen that we know currently that will cross the placental barrier. So it will actually infect unborn children and induce premature birth. And so this is the pathogen that when if you have been pregnant, or you know anyone that has been pregnant, why you or other people have been told like you can't eat cold cuts, you can't eat this, you can't eat that. It, in many ways revolves around this particular pathogen. It is a really, really nasty pathogen in terms of gastrointestinal infection, but it also has a profound danger for unborn children. So, with that, diseases caused by Listeria. So for all of us in this room that are normal, healthy folks, you'll get flu-like symptoms and you'll get really bad diarrhea. And if you were immunocompromised, one of the things that would emerge for you with a Listeria infection is that it is really particularly bad at inducing meningitis in immunocompromised adults. So if you have HIV, for example, Listeria is bad news for you. And if you are pregnant, Listeria can be really, really dangerous, not only just for you in terms of discomfort, but also for an unborn child. So, with that sort of happy story, I will leave us to finish up talking about how Listeria contrasts to Salmonella for the next time. Before we all pack up and run away, are there any questions? Oh, is one quicker than the other? Generally speaking, I would argue that the zipper mechanism. Yeah. Good question. Sorry? Oh, yes. Exams. Right. Be nice to each other when you run up here and tear these things away. Alright, so here are the written exams, and here are the verbal exams.
What? No.
Yeah.
Yeah.